Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm excited to be here today with Melissa Haendel, who is currently the Director of Translational Informatics and Precision Health and a Sarah Graham Keenan Distinguished Professor in the Department of Genetics at the University of North Carolina at Chapel Hill, which is where I did my undergrad as well. So we spent a lot of time in some of the same buildings, I think. And she also leads the Translational and Integrative Sciences Lab. Melissa, if you haven't read her work before, is really an expert in data integration, precision medicine, and genomic health, in particular with a focus on diagnosis of rare diseases, advancing public health surveillance, and really is one of the key people in data infrastructure within this field, in particular in the US, but really nationally and internationally renowned. So she's co-founder of the Monarch Initiative, which I used a lot of the work, human phenotype ontology, and some of these others when I was a PhD student. So I'm just really excited to have this conversation. We met a while ago, through some discussions around national scale rare disease diagnostics initiatives in the US. And I'm just really excited to have this conversation. So Melissa, thank you for joining us.
So great to be here. Thank you so much for having me.
So every hero has an origin story, and yours is a particularly interesting one I want to start with, which is you found your first professor role on Craigslist, I understand. So if you could share that story, I think I don't know the story, so I'm really interested to hear it. I know the audience would be as well.
So I had done several postdoctoral fellowships in basic research doing bench science. And I was going to have a baby and had been moonlighting at the zebrafish database for extra money at the time, where I was responsible for developing the first ontologies for anatomy and phenotype characterization of the zebrafish. And at the time that's where I really became an ontologist. And when I had the baby, I went to work at the zebrafish database full time because I didn't want to work on aflatoxin at the time. And I ended up developing a method in my spare time with a colleague of mine, Chris Mungle, who co-founded the Monarch Initiative with me, where we were able to compare phenotypic attributes of the zebrafish to human diseases for which we did not know the molecular mechanism, and then reveal the candidate mechanisms based on that phenotype similarity. And that method today is still used in our flagship diagnostic tool, the Exomizer tool, which is led by Damien Smedley at QMUL in London. But at the time, I was really interested in, and I had sort of, gotten off the track of becoming a faculty member until I saw this ad on Craigslist. I was actually looking for a truck. and I happened upon this ad for an ontologist. And, especially at the time, there were not exactly very many ontologists in the country, let alone any other ones that I had ever heard of in Oregon. So I applied. It turns out they had already hired somebody. And I said, well, just please, once they're in place, then I would love to get together because you're probably the only other ontologist in the state. And so then they're like, well, why don't you just send us your CV? And so I sent them my CV, and then they said, can you come up tomorrow? And I went and had an interview, and they ended up hiring both of us. And so that's how I got my first faculty job at Oregon Health and Science University in Portland.
That's amazing. Did you get the truck as well, or did that?
I did not get the truck.
That's a priority. For people who aren't familiar with Monarch and to whom ontologist is a word they might struggle to spell, I'd love if you could talk a little bit about the role that ontologies play in medical research more broadly, but rare disease in particular, where you've had a huge impact.
Yeah, so I think, one of the really exciting things about that original, kind of effort to build that algorithm and characterize the phenotypes of different species, was really description logics. It's basically, pretty foundational logic properties, like if A is part of B and B is part of C, then A is part of C is a transitive property. Well, by structuring data in that way, in an ontology, which is a sort of multi-hierarchy classification that a computational reasoner can then, do that kind of logical reasoning over, we can actually infer information that's not otherwise apparent. And then if we can structure phenotypic information that essentially allows us to do that kind of computational comparison across species and within species, then we can actually find non-exact matches. So for example, if a zebrafish has, 3 different phenotypic characterizations, like maybe there's a, callosal agenesis where the corpus callosum neurons don't cross the brain midline. There's, tail degeneration, I'm just making this up, and, cardiomyopathy, then those terms don't have to exactly match what we might see in a human in order to reveal the underlying mechanism. So we might only know that there's a heart abnormality, or that there's a tissue regeneration problem, or that there's, a neuronal growth problem. So they don't have to be exactly the same in order to have the reasoner tell us that they're similar. And by doing that, and combining with other data, we can actually kind of create these hypotheses for what the underlying molecular mechanism, variant, or gene might be that's responsible for causing those changes.
Yeah, no, when I first interacted with the human phenotype ontology, the application was looking at kids in the UK that had severe developmental disorders. And we were looking at, a large group of around 10,000 families where we had genetic data, but then we also had phenotypic descriptions of the child from the child's doctor, and trying to cluster these in that multidimensional space to look for, to your point, 2 children that wouldn't have been described as precisely the same by the doctors, but were actually very close in that ontology, and then also happened to share a genetic variant. And you then chase down the hypothesis of, are we looking at a new syndrome here? I'd love if you could talk a little bit about, we have the ontological backbone and descriptors to do probably some really amazing rare disease diagnostics these days, and use new tools like machine learning and AI to look across health records and other kinds of systems. But that clearly isn't matching up to reality. We still have diagnostic odysseys that patients go through in every country. If you could just talk a little bit about where the big gaps between theory and practice are right now, I think it'd be really interesting to just understand, where could we be doing a lot better, but we just don't have the tools or infrastructure in place quite yet.
So yeah, those are a lot of questions in there. Let's see. So I think,
good luck with that one. Yeah, take it whatever direction you like.
Yeah, historically, so the human phenotype ontology, which was originated by my colleague Peter Robinson, he and Chris Mongo at Lawrence Berkeley National Lab and myself really created the underlying logic that allowed this kind of phenotype comparisons, both within human patients as well as across species. But we've been capturing that information clinically outside of the electronic health record, because the characterization that one might do about individual patients isn't really captured in structured data very well. So if you happen to have like spider-shaped fingers, or your eyes are widely spaced, these are not things that you can bill for. They're not things that are correctable or druggable. And so they're often, if they're recorded at all, they'd be in the free text notes. And so, a lot of what ends up happening historically is that information will be collected outside the electronic health record for the purposes of doing the diagnostics. And that takes a lot of effort and specialty expertise in clinics that not everyone has access to. Nowadays, we also have the ability to run large language models over the notes. And so we can actually combine the ontologies together with the notes to extract more information that way. We also have the ability to potentially get information directly from patients using similar technologies with voice to text, or just, free text that the patients write. And we have seen, and this is not published, but there's a big difference between what the clinician characterizes and what the patient characterizes. And oftentimes they're not even overlapping. Wow. So information about what the patient is saying their problems are is not actually getting into the medical record, but holds the keys to a lot of the diagnostic capabilities when we're thinking about leveraging all the data from humans, all the data that we have from these different model organisms to help us understand what the underlying mechanism is. If we're relying only on what's in the electronic health record, especially only in the structured data, that's a big problem. So that's one of the big gaps, is just really capturing, more granular details out from the individual patients, as well as the free text in the clinical encounter context. The other thing that's really missing, that's a huge problem, is that most rare diseases do not have a standardized code
Right.
in the International Classification of Diseases or in the SNOMED. And so the community as a whole has really come together to reconcile all the rare disease definitions and identifiers across many different systems in a new ontology called Mondo. And we've been working really hard on trying to get those codes into electronic health record systems so that we can just do a simple query of like, do we have any patients that have a given rare disease? Turns out to be a very challenging query when there's no code for that. There's no diagnosis recorded for that. And then finally, I will say that even for those that do have codes, we often code wrongly in the context of clinical care because the clinician thinks, for one reason or another, that that patient will receive the right care, like for billing purposes, or to make sure that it's allowable under their insurance to receive a certain drug, that they will miscode on purpose the actual diagnosis in order to provide the best possible care for their patients. And so we are polluting our data a priori, and we need to stop doing that. We need to have a different way of recording the actual diagnosis with whatever coding system we have in hand accurately to the best of our abilities, and separate that from the billing context.
Yeah, that is really interesting. On the first point that you made about patient self-phenotyping, I understand you've got a paper coming out in the near future on this. I'm curious about whether you've learned how much signal is there, and also are there systematic areas where there are dark zones of the medical records and the patients actually have really powerful information that we're missing systematically. I'd love to hear what you've learned about that.
So what we've been doing in this paper is we translated the human phenotype ontology into layperson terms.
Yeah.
And about a third of the terms that are in the human phenotype ontology are translatable. So some of the terms are very technical, like laboratory, kinds of terms, and are not really like, there's no layperson equivalent, but like, terms like widely spaced eyes are something that a patient can understand, but they may not know what hypertelorism is, right? And so by translating those terms into layperson, we can allow patients to self-describe using the same terminology that we're using for the diagnostic workup. And so it's really that translation that is so exciting. And so we published that a number of years ago, but in this recent paper, we were able to simulate if a patient completed their own self-characterization perfectly, but using only that limited subset of terms that were available to them, how well would we be able to use that information to help diagnose them? And it turns out that works really, really well. That even with only a third of the ontology, we actually get pretty close. And so it really sort of, begs the need to actually have tools that allow structured collection of information from patients to be merged together in a collaborative fashion with the clinician, diagnostician, who's bringing different kinds of content and expertise to the table. And so then we tested it in a kind of early version of a tool. And we also did some permutations with like, I had a headache that day. It's not related to my rare disease because there's a real life, right? And it still was pretty robust in the face of that. But then we tested it in an actual application with patients and compared it to a standardized survey that just sort of is a whole body survey. And the patients liked the survey better than auto-completing on very specific terms. It's more laborious. And they didn't do so well in the use of it. So they did not really meet what our expectations in terms of what we thought they might be able to do if they had access to this layperson terminology. So I think we're kind of a little bit back to the drawing board, but this was at the very early times when LLMs were not yet prolific when we did the actual clinical evaluation. And so now with LLMs, really so prolific, I think we have a great opportunity to extract that kind of information directly from patients in ways that we just never thought possible before the advent of LLMs. So we're kind of thinking about how do we redesign that collection strategy, and then also thinking about how do we make it more dynamic so that it's using the prompts that we have in the LLMs, but also the underlying logic of the ontologies to say, if a patient says, well, I have this, and the clinician says, well, we should run this lab, then it should prompt the patient to ask another question about a potentially related phenotype. So a lot of these rare diseases are multi-system, and they get missed clinically because you're seeing a specialist. So if you have an eye disease phenotype that's brought you into the clinic because you're having visual problems, they might not know to look for, kidney dysfunction, but the ontologies and the LLMs know that. And so if we can actually use those kinds of prompts in a dynamic fashion between the patient and the clinician diagnostician, everybody, wins, and we learn more about the conditions as we go as well.
Yeah, that's really interesting. I have a personal anecdote that I think is relevant for this. I've shared on this podcast many times. We have a rare genetic kidney disease in my family that was recently maybe a year and a half ago, they discovered the genetic variant behind it. Something that we've always known is that the family members who go on to get a kidney transplant get high blood pressure very early on in life. But it's not we don't have proteinuria or any of the other like kidney disease hallmarks. So there are some of these clues that even kidney doctors don't really ask about, and it doesn't come up. But like for our family, that's quite an interesting, like everybody gets very high blood pressure, even if they run marathons or whatever in their mid-20s. And so there's some of these things, especially if it's a genetic disease or a syndrome, that you kind of need to connect the dots across the tree in a way that one subspecialty doesn't always know how to connect, right?
Yep, absolutely. That's the classic story. One of the exciting things about UNC Health that, we're kind of really building on since I've come there is they do screening for familial hypercholesterolemia, which is a, elevated cholesterol due to a specific variant. Well, lots of people have elevated cholesterol, but mostly not due to the specific variant. And it's very intractable to standard treatment and can really, increase the risk of death quite substantially. And so it's really important that we tease out those individual patients from the other ones, right? And so there's now a screening tool that allows clinicians that just looks at the pattern of cholesterol measurements over time and a few other clinical lab values and says, hey, you need to be sequenced for this variant. And it's like, we're getting like 80% more patients diagnosed now with this. I mean, it's just an incredible story of how just by looking at those kinds of patterns, we can actually like really affect the outcomes of these patients' lives, and it's that same kind of multi-system, pattern kind of assessment that's so lacking most of the time.
Yeah, so you have a lot of academic work, I think, tackling this problem, but you also are working on a company, Alamya Health, which I think is going after this problem in a similar way, well, from a different angle, but going after the same problem, working on basically making it cheaper and easier to have a clinically approved diagnostic lab in places where traditionally you wouldn't find them. More, I think more, rural environments or countries where you don't necessarily have the infrastructure that you have at UNC or Broad or somewhere like that. I'd love if you could talk a little bit about how you can see this problem being solved from the community level or from more of a ground up approach versus the top down that isn't working, right? It's not getting to everybody.
Yeah, it's a really important point. So my colleague, Neil Burkle, who helped co-found the Undiagnosed Disease Program's clinical lab at NIH at the time, and that was actually, we first met because he had learned about what the Monarch Initiative was doing with our model organism data. And we really helped build that early pipeline for using the model organism data to help diagnose patients. And it really worked. And it was really our sort of launch as a consortium. And so we go way back to those early days when I was first a faculty member at OHSU. So he's the president of Alamya. And what he said back in that day was like, if the undiagnosed disease program is successful, it will become obsolete because we will put genomic medicine will become medicine. We will just simply have genomic medicine be part of the everyday practice of medicine. And maybe that was a little optimistic at the time, but
Directionally correct, though. Yeah, we're getting there.
It will come eventually. It's just not as fast as I think we had all hoped that it would. And so part of why Alamya exists, it kind of harks back to that early premise, is that genomic medicine should be available to everyone, and it really should be part of general practice of medicine. But it turns out that's not happening, nearly fast enough or well enough. And I think even people think, oh, well, everyone has access to genomic diagnostics. Well, the answer is they don't, and especially not the best genomic diagnostics. So, we have some really great cutting edge technologies, multimodal AI-based analytics at Alamya. There are several other companies that are doing related things. There's some really great cutting edge technologies there that are really not accessible to the general public. They are accessible to people who are seeing very specialty clinics. And so the goal of Alamya Health is to bring that kind of to make that kind of diagnostic testing inexpensive and accessible. And one of the ways that we're really making it accessible is not only we're not just a diagnostic testing company, of which there are very many, not to say that they don't have an important role to play, because they're absolutely critical in the ecosystem. But the unique thing about Alamya Health is that, we aim to put genomic testing into facilities that currently don't have them, to really make genomic testing part of the everyday practice of medicine, and really bring precision medicine to various kinds of places like county hospitals, safety net hospitals, resource poor countries, and teach the people who are in those organizations how to set up their own physical diagnostic infrastructure and how to interpret the outcomes of the test, so that they can actually build it into their own pipelines and have a clinically certified lab in their own environments. And so, for example, we've been working with the state of Louisiana to put one of our first implementations into the state, to create a diagnostic lab in Louisiana. And in Louisiana, there's less than, I believe less than 10 genetic counselors, and none in the north part of the state, right? So it's this huge just expertise gap. And so if we can, kind of train the trainer, so to speak, then we can actually kind of deliver on that promise that Neil had wanted for the Undiagnosed Disease program so many years ago, of actually bringing genomic medicine to everybody.
How do you think about resolving the potential paradox of what sounds like what you're doing is very advanced technology? You have whole genome sequencing, also reflex testing, I think, to transcriptome and methylation. So it is the Rolls Royce of technology, but deploying it in a way that needs to be low cost and streamlined. How do you think about, achieving those two things together?
Well, I know it sounds silly, but just having one test instead of dozens makes a big difference.
Right.
So right now, especially the way insurance is, they want you to have this test, and then this test, and then this test, and then this test. And every time you do another test, it adds time and money. And the tests don't build on each other very well. They're often through different companies or different technologies. And so part of our strategy is just one test. And mostly we're not covered by insurance. So, we're the company of last resort. And we get the hardest possible cases that have failed all the other testing. And that's a really exciting and special place to be, especially with our early phase company, because we're really helping families that have gone a very long time without having a diagnosis. And so it feels really good to be able to help those kinds of families. But because we don't have the burdens of insurance saying you have to do all this sequential testing, we can do it, and the technology is such that we can do it in one test. The technology is also not large. It doesn't take a whole room full of machines to do this anymore. So it really comes down to single test without insurance being involved, and small, straightforward technologies. And then that's where the analytics and AI part comes in, because we have this multimodal strategy. That, together with the sort of single test, really makes it less expensive and faster, and also something that we can give to these organizations. Whereas you can't give an organization 25 different tests that all have different insurance needs and different infrastructure needs and different expertise needs. So it just makes it much more feasible and scalable for those kinds of, resource or expertise poor environments.
Yeah, I think that's a great point, because the administration time to go through that multiple rounds of tests, also the interpretation time to go through all those multiple rounds of tests. If you can collapse that down into a single test, and you can also collapse the interpretation costs through AI and other kind of automation, I think you're right that you can get to a world where you don't have to choose between cost and comprehensive testing, right? There's definitely a path I can see where actually you cut out the huge waste, and that's not even taking into account the opportunity costs of these families having to go through years of diagnostic odyssey, right? Which is hugely costly and not factored in, I think, really accurately in our current system.
Yeah, I would love to have more health economics folks help us with these problems where, what is the cost of not having a diagnosis? And there have been some analyses on this in terms of like healthcare costs, and in some cases we know that it costs a lot more not having a diagnosis. And in other cases, we know it costs more having a diagnosis. It just depends on the individual patient. But we don't ever measure the cost to society, the cost to the families, the cost of just quality of life issues, the sort of social costs, other like, if it's a pediatric case, like the extra support staff that might be needed in school or in transportation or in other caregiving activities. Like, we don't really know how much those costs are to, in terms of a societal or familial burden. And I think that it's important to take those things into account. We really stopped doing economics right at that point of like, what does it cost the hospital system or the insurance? Not,
Right.
and not thinking about the broader economic effects.
Yeah. You've been at the center of some pretty major national scale data sharing and aggregation analysis initiatives like the National COVID Cohort Collaborative and all of us as well, the major genomics program. I'm really curious to get your perspective on what you've learned being at that frontier, and what's the hardest thing about doing national scale research in the US? Is it about getting access to the data in the first place? Is it about harmonizing across centers? I'd love to just get a sense of what is challenging about the US compared to some other systems people might be familiar with, like the UK or areas of Scandinavia where we're smaller and less diverse populations, but maybe more national infrastructure.
Yeah, I wish we could have shiny things like that in the United States. And I'm actually writing a paper right now, which will hopefully be accepted soon, but that's about how can we make real world data a public utility like water or electricity, which it kind of is in some of those other countries, right?
Yeah.
Like, it's not rocket science to say, we should use all the data we have about you as a person and all the data we have about other people to decide what to do about you and your problems, right? It's kind of like a no-brainer, but we don't really do that in medicine. And so we'll come back to that in a second. But when the pandemic happened, I happened to be leading the coordinating center for the Centers for Translational Science award sites, which is 60 academic medical centers. And this program, the Center for Data to Health, had been tasked with developing governance and infrastructure to help academic medical centers share and harmonize their data for doing analytics. Well, it wasn't going very well because it turns out there's not really a lot of incentives in the United States for organizations to kind of share at that scale. They'll share readily for specific projects, like if you've got, a clinical trial or something like this, very, very, very willing to share for that purpose. But there's not really like a good incentive or a good infrastructure for having done so. And so we were making fairly slow progress in doing that until the pandemic happened. And we said, hey, what we really don't have is any kind of centralized data where we should be using all the data we have about all the people in order to decide what to do about each person, especially in the face of this new disease that was COVID-19. And we didn't really know what all of the, ameliorating factors might be, what drugs were helping outcomes, what drugs were hindering outcomes, what comorbidities put you at highest risk of severe outcomes, these kinds of things. And so we really, what was the most special thing about that time was the very unique public-private government partnership. So the community really came together and said, here's how we want to share data and how we want to have the data be used. The government was the steward of the data, and the technology partners, in particular, Datavant, Axel Informatics, and Palantir, were able to actually realize the physical constraints that were needed to protect this very sensitive data. And so that unique partnership was really what made that happen. And unfortunately, we no longer have that. And so that's part of why I'm writing this paper about, the need for real world data to be a public utility that has, like public utilities, have, citizens involved in the decision making around, how those data are collected and used, but also need those commercial partnerships to have the right infrastructure, and they need government regulation as opposed to government ownership. The government does not own the electrical grid, but they do regulate it, for example. So I think we need a model that's similar to that. The All of Us research program has been a very different kind of program and a very interesting opportunity to really kind of bring to fruition this idea that, and I like to call it my very technical term of putting the patient back together again, which is basically in the research space, we often take individual data types about a given patient and we ship them off to their individual data repository. So the genomic data will go over here, the imaging data will go over there, the clinical data will go over there. And we never put them back together again. So we're not really doing the multimodal analytics that we should be doing in order to really realize the dream of precision medicine. And of particular importance is the lack of integration of geospatial and geospatially temporalized data like environmental and economic data. So, really understanding where a person has lived and linking those environmental factors and those social factors to their sort of, patient history over time, and then understanding how those influences positively or negatively affect clinical outcomes. And then how do the genetics influence those outcomes as well? And so the whole genome sequencing that we have in the All of Us is really amazing resource. And then the All of Us program also has wearables and lots of rich survey data. They're starting to collect eye images. We have nutrition biomarkers in biosamples. It's a really, really incredibly rich data set and critically important to maintain this cohort. So I hope that our US government is invested in that, because it's a very unique opportunity in the United States in particular, especially in the face of not having that centralized EHR infrastructure. And so we've been able to, as part of the Center for Linkage and Acquisition of Data, or CLAD, be able to sort of really focus on the linkage of data across different sources. So different claims data for insurance claims really complements what we see in the electronic health record data.
Right.
So for example, just because you got prescribed a drug doesn't mean you filled the prescription, which is more likely that you're actually taking the drug if you actually filled it. That doesn't mean that we know you actually took it, but it's a lot better approximation. Those sorts of things can really help fill some of those gaps. And then finally, kind of for the first time, tapping into national care networks for exchange. So there are laws in the US about sharing data for clinical purposes. So if you're seen at one hospital, there needs to be a mechanism to get data from that hospital to another one in order to help support the care of you at that second hospital. So we, for the first time, have tapped into that for research purposes via a partnership with the eHealth Exchange Health Information Network. So we're the first research organization to participate. And so we are now pulling data based on participant authorization for the purposes of research through that network. So we can go out and find all the EHR data about you, wherever it might be, through this network, and then pull it back in and combine it with the data we already have about each individual participant. So this is a really exciting time where we're sort of really laying some of the groundwork for how do we kind of leverage and combine the things that are already in place for that sort of, dream of, real world data as public utility, for how we can actually get there. So that was a long answer.
No, it's great. I mean, it's amazing how much progress you're making on this. To get access to that data through, I think, as eHealth Exchange you mentioned, who do you have to talk to get that exception made, or is there a pathway for researchers to actually do this, but people just don't realize it? How do you, because I think that's a big unlock for a lot of people who want to do this kind of research.
Yeah, and so maybe there's two answers to that question. So the first one is that the CLAD, the Center for Linkage and Acquisition of Data, was on National Institutes of Health investment for national infrastructure. And so now that we've actually got it up and running, and that's working with one health information network, which does not cover every American, it's only a subset.
Yep.
It is available to other research programs. So we don't want to have people have to redo that. It was expensive investment. And there was a lot of legal and regulatory work, as well as just, data architecture and infrastructure work. So we're really kind of welcome, requests from other research programs to leverage the infrastructure and share the cost of that infrastructure. We're also working as part of TEFCA, which is the new policy that says all health information networks need to be exchangeable across one another. So now we can start working with other health information networks that, have access to different patient populations. And it's very fractal. So the health information network then gets data from their health information exchanges, which tend to be regional, sometimes they're state. And then they interact with their health provider organization. So the health provider organizations send data to the health information exchange, sends data to the health information network, sends data to the Center for Linkage and Acquisition of Data, that sends data to wherever the research, environment is that we're going to be partnering with. And so it's a two-way street. We have to be able to send the authorizations, the consent forms, to the individual all the way down the line to those individual providing organizations.
Yeah.
So we're now starting to work on, just in our next phase of development that's just started, on tapping into other health information networks as part of the umbrella under TEFCA. And so this is really exciting, because if we could actually get this to work for all TEFCA HINs, then we would have we would actually have that nationally for any research program.
That's fascinating. And you can just imagine the questions that are just impossible to answer today that you could start to answer with that kind of integration, right? And the way you described it is, it sounds a lot like water flowing through a pipe to get to its destination.
Yes. Yes, exactly.
With you, it should be a utility. Yeah. As we,
one thing though to note though is that it's now that we're doing this, the data quality that's needed for care and sort of just attributes of the data is different than what we need for research to some extent. And, we're realizing things that we just never would have thought about as we were getting these data. So for example, we might only have a few years worth of vaccine information, or the vital signs might be dropped by a given HIE because they just don't need them for some reason, or they didn't notice that they were missing because they just weren't the thing. When you're recalling somebody's electronic health record data, unless you're particularly interested in their vaccine history, going way back, it's just not what's needed for the immediate care purpose. And so those kinds of things are much more important for research, where we really want that richer, more accurate longitudinal history and just cleaner data than one might need at the point of care for care purposes. But at the same time, we're kind of, by looking at it through this lens, everyone's data is going to improve, right? And that also means care will improve, even though it's not as, that information is maybe not as urgently needed at the point of care. It still will eventually improve care and precision medicine.
Yeah, and like the way I'm thinking about it in the rare disease world is, you've got on one extreme end of the XY axis in the bottom right corner, you've got randomized control trials where you have very small number of patients, very rigorous control of the data. Somewhere a little bit closer to that, but more in the real world space, is natural history studies, but still very controlled ways of understanding, in the absence of treatment, how does this group of patients change over time. And then in the top left of this, very, very large data, but very kind of, data quality, data missingness. You have this vast real world data that could help you answer some similar questions in natural history studies, like what does the patient's journey look like, but you have to tackle this, this missing data or data quality problems. But there's really interesting science to be done in between those two, right? Of where you can get the volume of a national scale infrastructure and find a way to impute or clean up that data in some way, right?
Absolutely.
As we wrap here, are there projects or things that you're particularly excited about over the next year or two that you want to share with people, or is there anything people can help you with? Is this something you're looking for that you want to make a shout out to collaborators or potential partners?
Well, I think one of the most exciting things for me lately is our Mondo disease ontology work, where we're really working together across all these different organizations to reconcile our rare disease definitions and knowledge, and bringing them all together to sort of help have more shared definitions. And this really came about because in our diagnostic work, we realized that the definition of that diagnosis was different depending on which country you were in, or which data source you were using. And we can't have our diagnostic tools, failing or giving different answers just because we're using, different knowledge sources.
Yeah.
So the community's been working together for about 5 years now to realize this Mondo ontology. And it's not meant to replace any of the original sources. It's really just a reconciliation and computational infrastructure layer. And recently we've partnered with Alamya Health, which is a terminology service provider for Epic and other electronic health record and clinical data systems. And they've just been a wonderful partner for us in helping us deliver the Mondo rare disease codes into healthcare implementations. And so this fall, we are going live with the first large set of rare disease codes in Mondo into healthcare in the United States. And so could not be more excited about this because, as I mentioned earlier, there's, you can't even query for, 80% of rare diseases don't have ICD codes. And so this opportunity to really help, diagnosticians and general practitioners be able to actually document what specific rare disease does each person have, that really stimulates research downstream and helps compare patients across healthcare systems and is really going to help improve the overall landscape of, rare disease public health surveillance and diagnostics, as well as clinical, identifying patients for trials and other kinds of observational research. And so what I would really love for the audience who's watching, if you have opportunities to engage your healthcare system, please tell them to ask for IMO Core and the Mondo rare disease codes and help us test it. Help us make sure that it's working for you and your environment so that we can, do a better job of delivering rare disease coding systems into practice and then also how we use those in downstream research systems as well. So that's our really big push right now.
Amazing. I do not know how you do all these things that you do, but I'm very glad that you're out there doing it. So thank you for sharing all this with us. I learned a huge amount, as always, when I talk to you, and I know the audience did too.
Thank you so much. It's really an honor to be here. I'm so grateful for you guys doing what you're doing, and I'm looking forward for our groups to work together more. So thank you so much for having me.
Yeah, glad to help spread the word. Thanks, everybody, as always, for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.